So welcome everybody to the March 2026 edition of the Malaria Gen Journal Club. This is our first journal club back for the year and also our first from our new home at the Liverpool School of Tropical Medicine. And my name's Nina White. I'm a data scientist within the Malaria Gen Parasite team. This is my first time chairing one of these meetings and our first time using Teams for it. So fingers crossed that everything runs smoothly, but do bear with me if there are any small hiccups. Just a little bit of housekeeping as well. If you could kindly keep yourself muted while our speaker presents. There is a chat and a Q&A function which hopefully you can see, and do please post any questions in there for Ashley during her talk. And we will have time for a Q&A session at the end where there'll be the opportunity to raise your hand, unmute and ask a question directly if you like, or I can read your question aloud too. So now, Kazay, we are delighted to welcome Dr. Ashley Osborne for today's talk, and she'll be presenting her work using genomics to decode Ethiopia's unique and structurally divergent Plasmodium falciparum populations. A little bit about Ashley. Dr. Ashley Osborne is a molecular epidemiologist with a background in molecular biology and malaria parasite genomics. Her research focuses on developing innovative molecular surveillance tools to uncover the hidden biology and epidemiology of both Plasmodium vivax and Plasmodium falciparum infections. She works closely with collaborators in Ethiopia and within the Asia-Pacific region to strengthen in-country genomic capacity for monitoring transmission dynamics and anti-malarial drug resistance too. Dr. Osborne is a molecular epidemiologist at the Menzies School of Health Research and contributes to malaria surveillance initiatives within the Australian Centre of Research Excellence in Malaria Elimination. So that's everything from me. Ashley, if you are happy to, please go ahead.
Thank you for the introduction. I'm just going to quickly share my screen. Is that okay?
All good.
Perfect. So hello, everyone, and thank you for joining today and taking interest in this piece of work that myself and many others have put a lot of hard work into. And a big thank you to Malaria Gen for inviting me to present this with all of you. And to jump in, so I've already had a lovely introduction from Nina, but we'll be talking about Ethiopia's Plasmodium falciparum populations and using genomics to explore the nuances of these population dynamics in this complex environment. So I'm going to start off with a bit of regionally specific insight into the current malaria situation in Ethiopia and some of the setbacks that it's unfortunately been facing on its journey towards malaria elimination. So unfortunately, in recent years, we have witnessed quite a dramatic resurgence in malaria case counts within the country. In 2019, Ethiopia had its lowest ever case count at approximately 900,000 cases, but in 2024, there was estimated to have been over 12 million cases. So this has been attributed to a combination of factors, including the geopolitical climate and internal conflicts and economic restraints in the country, as well as the emergence and spread of Anopheles stevensii from South Asia into the Horn of Africa, which has been acting as a very competent vector in urban environments. This has been combined with the evolution and spread of drug resistance in both malaria parasites and vector resistance to insecticides, as well as climate change, which has been allowing for a more sustained and year-round transmission in regions that previously only had seasonal transmission. So although Ethiopia had previously been on track to meet its global technical strategy target of 95% reduction in malaria by 2025, this goal is unfortunately no longer achievable and highlights the need for renewed intervention strategies and technical advancements in the field. So perhaps one of the biggest concerns we've been facing in Ethiopia has been the evolution and emergence of molecular markers associated with susceptibility to artemisinin, and this has been slowly spreading across the region. Interestingly, these resistant parasite lineages don't appear to be evolved separately from the strains initially reported in the greater Mekong subregion, where reduced susceptibility to artemisinin was first observed and characterized. So the notable R622I variant on the Kelch13 gene was first reported in northern Ethiopia before it has since steadily spread further south along both the border with Sudan and South Sudan. And there's some preliminary evidence that suggests this variant might readily be arising on parasites that carry HRP2 and 3 deletions, which would compromise our ability to detect these resistant lineages using standard RDTs. In addition to the spread of these markers, there's been concerning reports in the region of delayed parasite clearance following treatment with artemether lumefantrine, which is currently the first-line treatment across much of sub-Saharan Africa for falciparum. And then in addition to artemisinin-resistant markers spreading, co-circulation of falciparum in vivax in the Horn of Africa complicates our ability to detect and treat malaria infections and introduces quite interesting nuanced drug pressures compared to other regions across the continent. So although chloroquine is no longer used for the treatment of uncomplicated falciparum malaria, it remains the first-line treatment for vivax malaria in Ethiopia. And there's actually been reports that some regions are experiencing an 11-fold increase in vivax cases since 2018. And so chloroquine's extended half-life and the ability for misdiagnosis can often result in persistent drug concentrations in an individual's bloodstream that can inadvertently maintain significant chloroquine pressure on falciparum populations. And although we're not using chloroquine to treat falciparum, this may potentially be selecting for parasites that readily uptake adaptive drug resistance variants. And so our main objective behind generating whole genome sequencing data from Ethiopia was to advance our understanding of the population structure of these parasites and address the limited availability of genomic data in the region. So to date, there had only been about 26 publicly available whole genome samples from Ethiopia. And analysis of these samples that were collected in 2013 highlighted what appeared to be rather unique and structurally divergent lineages from the rest of sub-Saharan Africa. And however, this small sample size precluded the authors from drawing any large-scale deductions from this data set. And the largest molecular investigation in Ethiopia so far has been using molecular inversion probe genotyping, which has generated some really important insights into parasite connectivity across the country and drug resistance distribution. However, the MIP dataset, the MIP genotyping doesn't have the resolution for large-scale haplotype-based analyses, which is used to monitor genome pressure, including regions under active selection. So fortunately, we had access to a data set that allowed us to generate whole genome data for 163 isolates from Arbaminch, Ethiopia. Now, these were collected between 2017 and 2021, and we were able to combine this with the pre-existing Ethiopian isolates from 2013, as well as 27 isolates from Sudan and 801 samples from sub-Saharan Africa. So this resulted in a really nice and robust data set across the continent, with our in-country analysis consisting of 187 Ethiopian isolates and a continent-wide comparison of these 187 isolates, as well as 830 samples from the PF7 data set, so a little bit behind on the PF releases, with 13 African countries from across the continent. So we first looked at within host and population diversity by calculating the FWS metric per sample. So these metrics range from 0 and 1 per sample, with high FWS values greater than or equal to 0.95, often reflecting monoclonal infections or infections caused by a single parasite clone, while lower FWS values tend to indicate a more complex infection made up of multiple clones. So in general, you'll see lower FWS values observed in high transmission regions where diversity and outcrossing of parasites between populations is relatively high. And here we can quite clearly see that Ethiopian parasites have a tendency to be monoclonal with a mean FWS value of 0.95, compared to other regions that historically experience rather high and sustained transmissions, such as Malawi. Now, this is rather interesting because despite the resurgence of malaria in Ethiopia since 2019, and 105 samples from our data set being from 2021 in this estimate, Ethiopian parasites retained this low within host diversity, suggesting that not much opportunity for population outcrossing is available despite this increased transmission. And then when we were looking at the overall continent-wide data set, we have approximately half of all of our isolates were estimated to be polyclonal, while the remaining half were estimated to be monoclonal. So then another metric of determining population relatedness and genomic conservation over time is to estimate identity by descent, or IBD, between parasites. So closely related parasites tend to share large segments of their genomes with one another that have not undergone recombination when inherited from the most recent common ancestor. And so this results in relatively high levels of IBD, while unrelated parasites share very low levels of IBD as outcrossing occurs between generations with external genetic reservoirs. And so parasites in Ethiopia display these really high proportions of IBD and genomic sharing between 2013 and 2021, with a majority of isolates sharing at least 25% IBD, if not more. And so interestingly, there were 3 isolates from 2013 that were observed to share greater than 50% IBD with parasites from 2021, suggesting closely related lineages that have been persisting for nearly a decade. Sorry, I just got a notification. Is the signal okay?
Yes, I think it was just somebody waiting in the lobby.
Okay, perfect.
Right.
And so one way we can put this into perspective, since you're suggesting 50% or 25% IBD, and what does it really mean? Most isolates from other regions across sub-Saharan Africa exhibit very minimal levels of IBD, rarely exceeding 5%. So this is highlighting this really strong level of genetic conservation and restricted parasite gene pool in southern Ethiopia and the Horn of Africa Sudanese isolates that we have in this data set. And so to explore the impact of this continuous chloroquine pressure in the country, we mapped the prevalence of chloroquine-resistant associated haplotypes across the continent. And so in contrast to its slow decline elsewhere, the resistant CVIET haplotype and the K76T variant on the chloroquine resistance transporter gene were fixed in Ethiopian populations regardless of their year of collection. So extensive co-occurrence was observed between this CVIET variant haplotype, as well as variants like A220S and 326S and R371I. So approximately 99.6% of the parasites in our population that had data available for every position were carrying this combination of variants. And then in addition to variants on the chloroquine resistance transporter gene, we also noticed this S258L variant on the AAT1 gene, which was also fixed in our population. And this variant is believed to confer resistance to chloroquine, but at a fitness cost to the parasite. So this is suggesting that pressure on falciparum from chloroquine use against vivax is actually strong enough to warrant a fitness cost for falciparum parasites. And then, as I mentioned before, that although we're not using chloroquine to treat falciparum, there remains the possibility that pressures like these may permit the selection of parasites that readily evolve and maintain adaptive drug resistance variants beyond just those attributed to chloroquine and may have a permissible drug resistance genetic backbone. And then in addition to chloroquine, we looked at sulfadoxine pyrimethamine. So sulfadoxine pyrimethamine was discontinued as a first-line treatment of falciparum in Ethiopia in 2004 and was actually never implemented as intermittent preventative treatment in pregnancy or as seasonal malaria chemoprevention in the country. And so despite a prolonged period of absence of drug pressure, antifolate resistant markers persist at exceptionally high proportions in our data set, with the DHFR triple mutant haplotype observed in 90% of the population. And the susceptible wild type haplotype was actually absent from our population. And so similarly, the DHPS double mutant was observed in about 98% of our isolates, and only 2 of our isolates were the susceptible wild type haplotype. And so these proportions are comparable to or even exceed in some instances those in regions where SP is actively used as IPTP or SMC. And this persistent antifolate haplotypes may reflect either a low fitness cost to retaining these variants or propose that there's maybe other sources of selection that are maintaining pressure on them and the reason for our populations to retain these variants. So one of the examples of a secondary drug pressure could be coming from cotrimoxazole, which is an antibiotic that is similarly targeting a similar pathway as the antifolates and is commonly used as a bacterial prophylaxis and treatment in Ethiopia, although it's believed that this alone isn't likely to be providing enough pressure to keep these resistance markers in a population. So there may be more going on here than we are aware of. And so despite the recent emergence and spread of variants on Kelch 13 associated with reduced susceptibility to artemisinin, we did not actually observe any validated or candidate variants on Couch 13 in our data set. And so this is despite our parasite populations in South Ethiopia exhibiting high connectivity with data that was collected during the molecular inversion probe research that was done by Abebe Fola and his team. And so despite some of our samples sharing up to 50% IVD at those molecular inversion probe sites, we do not see that this variant has spread to our population just yet. But given the connectivity that we are seeing between these regions, there is a strong likelihood that more recent sampling in the region could identify parasites carrying these resistance markers, or that the variant itself may spread soon within the region. So it's important to keep an eye on this. And so an advantage of performing whole genome sequencing is our ability to screen samples for both known and novel structural variants, rather than just SNPs or deletions in specific regions. So we were able to look at the HRP2 and 3 genes, and the loss of one or both of HRP2 and 3 can impact the diagnostic sequence relevant to the rapid diagnostic tests and impact their ability to actually detect malaria infections. So our findings aligned with previous descriptions that HRP3 deletions were fairly widespread across the Horn of Africa, including Ethiopia. So we saw about 50% of our isolates from 2021 having this HRP3 deletion, up slightly from the data set in 2013, although that was a smaller data set. And previous reports had suggested that this HRP3 deletion tends to precede the subsequent loss of HRP2. But in this instance, none of our isolates were carrying both deletions. So all of our HRP3 deleted parasites that were collected in 2021, and they all had intact HRP3 genes. There's also some preliminary evidence to suggest that these HRP2 and 3 deletions might be linked with the subsequent acquisition of this R622I variant on Couch 13 that's been spreading across Ethiopia, but further work is needed to confirm whether or not this link actually exists. And so in addition to identifying deletions on HRP2 and 3, we were able to identify a small indel within the 5' UTR of MDR1 and about 40% of our Ethiopian isolates. And so we only had a data set of about 65% because we required there to be high depth in coverage across the entire region. And then this variant was also identified to be in approximately 32% of both East African and Central African isolates. And so, although it's an untranslated region, there is potential that this could be implicated in modulating some mechanism of drug resistance. So long-read sequencing would be beneficial to confirm the presence of this variant, as there's potential for transcriptional regulation and the modulation of resistance through MDR1. A similar activity or mechanism of action has been hypothesized for a deletion in vivax populations in Cambodia following the detection of a 3' UTR deletion. And interestingly, because this region is often not covered by targeted amplicon sequencing panels, which usually screen for known transcriptional variants, this variant may have been not being detected as readily and may have been missed or unexplored, as it's not generally looked at or sequenced in these more widespread amplicon panels. So another merit of having some whole genome data available to really explore this Ethiopian population. Another benefit of having this whole genome data versus more targeted or molecular inversion probe data is we're able to perform slightly more robust haplotype-based analyses using to identify signatures of selection across the genome. And so this is done by calculating baseline levels of heterozygosity across the genome to then identify regions or positions outside the normal range. And this is known as an IHS score. And so positive IHS scores in this instance were indicative of the ancestral allele or the reference allele being under selection, whereas a negative IHS score was representative of a newly derived allele under selection. And we had hypothesized that this prolonged artemisinin usage, partner drug, and residual chloroquine exposure would result in some temporal shift in signature over time. And we identified a number of genes under selection across the different years of collection, but they were predominantly linked with host immune evasion, cytoadherence proteins, and conserved exported proteins, so usually involved in invasion and immune evasion, so invasion of red blood cells and host immune evasion. We did identify a signal on DHPS in the 2013 population, as well as signals on the PFUT gene, which was identified in both the 2013 and 2017 populations. So this gene is hypothesized to be involved in resistance to both quinines and quinidine, although I don't believe the link has been quite confirmed yet. But we didn't observe, using this metric, we weren't able to observe high correlation between selection on known drug resistance markers. And so in addition to these IHS scores, we looked at regions exhibiting excessive proportions of IVD compared to the surrounding genome as another proxy for measuring genomic selection. So these regions are being retained despite multiple rounds of recombination and outcrossing with other parasites. And so these IVD-based scans revealed recurrent hotspots across validated drug resistance genes, including MDR1, CRT, DHFR, and DHPS throughout the different years of collection, as well as drug-resistant candidates genes that were also identified to have high proportions of IVD, including AAT1, AP2MU, and GCH1. So it is hypothesized that as well as being involved in chloroquine resistance, that AAT1 potentially plays a role in artemisinin resistance, whereas GCH1 may be implicated in antifolate resistance. And so to look more at the genetic differentiation and distinction of these populations versus the in-country analyses we were doing, we generated some principal component analyses of the samples from across sub-Saharan Africa to really showcase this high relatedness amongst sub-Saharan African isolates, despite the wide geographic proximity and the clear distinction between these populations and those populations within the Horn of Africa. And so here you can see quite nicely that the Horn of Africa is just really clustering very differently and distinctly from those sub-Saharan African populations. And so to further investigate these population dynamics, we carried out admixture analysis used from the Tessor software using the same data matrix as the principal component analysis. And so this Tessor analysis combines matrix factorization and spatial statistical analysis to provide ancestry estimates based on a least squares approximation. So using this approach, we identified 6 major ancestral components that were identified that broadly kind of align with the general geographic subregions within sub-Saharan Africa, which is West Africa, Central Africa, South Central Africa, the Horn of Africa, East Africa, and Southeast Africa. And this also aligned fairly well with historic transmission connectivity between the regions. The K2 and K5 ancestries corresponding to the Ethiopian and Sudanese isolates were rare elsewhere in Africa, kind of reinforcing the PCA and preliminary research suggesting these unique evolutionary trajectories when it comes to the Horn of Africa's parasite lineages, as well as the distinction between the human populations between the Horn of Africa and sub-Saharan Africa. IVD network analysis further supported this genetic distinction and lends support to the idea that the Ethiopian falciparum populations are likely genetically isolated reservoirs shaped by local drug pressure and ecological pressures rather than the continental gene flow. And despite their geographic proximity, Ethiopian and Sudanese falciparum populations exhibited these distinct ancestry signatures, with Ethiopia predominantly composed of this K2 ancestral component versus the K5 component, which made up the Sudanese isolates. And so at the time of this analysis, whole genome data for Sudan was limited, but a larger data set and broader analysis is needed to further support these trends and figure out what's going on here and what is driving this differentiation between these two populations that are geographically quite close. And I do believe more data has become available recently, so that would be really interesting to dive deeper into. And so in conclusion, I know I've glossed over some topics, but feel free to ask me some more in-depth questions at the Q&A session. But our analysis revealed that these falciparum populations in southern Ethiopia are highly structured and genetically constrained and shaped by these nuanced selection pressures. And this rather bottlenecked and cut off population in the Horn of Africa. And the fixation of these drug resistance haplotypes, despite the continued use of chloroquine and sulfadoxine pyrimethamine, together with this new novel MDR1 5' UTR indel, highlights the resilience and adaptability of these parasites. And then just the strong connectivity in northern Ethiopia and the signatures of selection at CRT and MDR1 indicate a relatively permissive genetic background that threaten the future spread of artemisinin resistance and the potential novel emergence for resistance in this region. So it's highly likely that this continued spread of artemisinin resistance from northern Ethiopia will eventually make its way into this population as well. And our findings underscore how unique epidemiological context of the Horn of Africa, marked by this lower transmission, limited recombination, and vivax co-endemicity, has produced an evolutionary trajectory distinct from other African falciparum populations. And the need for sustained genomic surveillance to tailor this local ecology and treatment practices will be essential to anticipate resistance evolution and guide effective malaria elimination strategies in the region, particularly in a time when resurgence is becoming quite problematic, and we will need some new technology to really get to terms with this. And so I really want to give a special thanks to the entire team at Menzies, in particular Sarah Auburn, Rick Price, and Kama, who have been really amazing with this piece of work and their ability to just be available and help and produce these amazing data sets, as well as the rest of the team who's really given their all to this project. And then I'd also like to give a special thank you to Richard and Shiyun, who made it possible for me to get through some really difficult structural variant analysis that I don't know I would have been able to figure out without them. Ahead of time.
Great. Yeah, thank you so much, Ashley. That was a really nice talk and covered a lot of ground there, and I think there's plenty for us to get into then for discussion. So just as a reminder to everyone, we've got time now for a Q&A. So if you have a question that you would like me to read aloud, please pop it in the Q&A or chat box and I'll try to get to it. Alternatively, I think hopefully you can raise your hand and I can try to unmute you if you'd like to ask it yourself. But to kick us off, there is a comment or a question in the chat already from Mr. B. Kathewera. So they say that the Ethiopia case shows how chloroquine use for P. vivax inadvertently sustains drug pressure on P. falciparum. In Malawi, I see a related challenge, but from the community side. Many people self-prescribe artemether lumefantrine for malaria-like symptoms, even when MRDT or MPS results are negative. This practice not only risks accelerating resistance, but also masks other febrile illnesses and undermines test and treat policies. How can we better engage communities to reduce self-prescribing patterns while ensuring access to appropriate care? I'd be interested to hear strategies others have tried in similar contexts. Ashley, do you have any thoughts there?
Yeah, I think it's really tricky, especially it's quite difficult to explain drug resistance in certain instances where I think we face similar in Ethiopia, where you're prescribing a medication for a febrile illness, and a lot of times the information gets misconstrued. But I don't think drug resistance itself is often described much at the community level. It tends to be kind of focused on at the higher project level where they're made aware of drug resistance, but maybe the information isn't made accessible to community members. So I think it'd be really interesting to see how we could actually make this sort of research and information more accessible beyond just the scientific research committee and the higher ups. Because yeah, I think the community, if they were given the same amount of knowledge base, would likely stop doing that.
I'll just let somebody in. Thank you. So yeah, I have a question as well, actually. So it was interesting, given the genetic connectivity between northern and southern Ethiopia, that there were actually no KELP13 mutations spotted. Do you have any hypotheses for why these mutations haven't spread yet? And just to follow up, you mentioned an opportunity for containment in the paper. And do you have thoughts on what this containment would require to be successful?
Yeah, on the first part, it's quite interesting to see the connectivity between the samples. I think the only caveat with that analysis is we're comparing the whole genome isolates to the myth isolates. So instead of having 120,000 SNPs to compare between those samples, we're looking at about 1,500. So there is possibility that those positions aren't capturing the true level of diversity, although I think it's highly likely that there is that connectivity given the population bottleneck, but that perhaps it just hasn't made its way down yet. And then our samples were collected from 2021, whereas I would not be surprised if 2023 or 2024 isolates did have this. Although the data set is 180 samples, only 120 about are from 2021. And as much as we think that's a lot of samples, your statistical power to capture a variant at 1% of the population is quite reduced at that level of screening. So it's very possible that we missed it, although we have spoken to some collaborators in the region who do think that Arborminch might be its own little quite isolated pot as well. So maybe there's not as much mixing as we think. And if there isn't as much mixing as we think, containment, I think, will have to rely on reducing transmission. I think, unfortunately, Ethiopia's moved slightly backwards on its elimination course and is more of a containment course at the moment. So if it was more of back in its elimination setting, we could do more nuanced tracking and tracing of cases. But I think at this point, it's just a matter of reducing transmission to as low as possible.
Okay. So we've got some more questions in the chat. I'll read them out now. So we've got Luciana Kohatsu says, excellent presentation. Do you know if a One Health approach being taken into the investigation of the genetic evolution of the parasite, for example, overlapping the genetic evolution with changes on weather patterns and vector distribution?
Yeah, I think the One Health, more nuanced approach to looking at this is really important. I think, unfortunately, it's been quite tricky to figure out how to do that sort of comparative analysis. I think there's been some studies that have done with the human genetics, particularly, that have been really interesting to show the dynamics that are happening between parasite and host evolution. So if that's happening, then there's almost certainly some sort of push and pull happening between parasite and vector. I'm not sure, I don't have much experience with vector distribution and weather patterns, so I'm not the person to ask on this, but I do think that there must be some role in play into what's happening genetically speaking between these two. But yeah, as we've seen, Anopheles stevensii in the region is almost certainly becoming problematic for this. So we can't just look at one versus the other. We have to approach both.
Yeah, absolutely. So another question from Sarah Volkman. Excellent work. Can you talk a bit more about the clonal parasite architecture in this population, given the low COI and high IBD? Do you see clonal clusters, and do these persist over time?
Yeah, so I think briefly touched upon it in that display of the parasites from the different years of collection. And so we do see clonal clusters, but they don't seem to necessarily be year dependent. Let me just see if I can go back. I'm not sure I can, but so I think the issue is because of the low COI in the population in general, it's quite tricky to know that if we actually have clonal clusters or if the genetics are just so bottlenecked that we don't have enough outcrossing to, because I think identity by descent is really useful in situations where you do have highly diverse populations versus not diverse. But in a situation like we're looking at, the IBD could potentially be misconstrued. So because there's no genetic information coming in, it looks like they might be clonal, but they might just not have any genetic information coming in to outcross with. So it's tricky, that one.
Thanks. So I have a similar question to the one I asked, but I think with a slight difference from Brian Odong. Thank you so much, Dr. Osborne, for your wonderful presentation. I'm still wondering why you didn't find the PF K13 variants in southern Ethiopia, given its presence in northern Kenya. So it's more about cross-border transmission than within country transmission here.
Yeah, so I think even though there's geographic proximity in northern Kenya, it's just a matter of, is there enough cross-border migration happening between those regions? I know there is movement across the border, but it's just a matter of whether or not that there's enough movement, enough individuals carrying those variants across the border and up specifically into Arbamint for us to detect it. And then, yeah, as I mentioned before, even if it's in the population at 1 or 2%, you need to sample thousands of samples to detect that. So we might have just missed it, and we might just need more statistical power. Yeah, I wouldn't be surprised if it was there.
So we have another question from Richard Pearson. Great talk, Ashley, very clear. Regarding the deletion in the 5' UTR of MDR1 you mentioned, this is at high frequency in East and Central Africa, but did you also assess this in samples from West Africa? Also, do you have a sense of whether this is increasing in frequency over time? And finally, do you have any plans for functional evaluation of this deletion?
So I did look at West Africa. I think I didn't include it in the presentation just because the number of samples that had full intact sequencing from West Africa in that region specifically was not very high. So I do have a frequency. It's just wasn't very statistically, or not statistically, it just, the sample size wasn't very large. I don't remember what the frequency is off the top hand, but I think it was comparable. I think fairly, the frequency or proportion of samples that had this was fairly standard across the continent, which was quite interesting. And then I don't have a sense of whether this is increasing over time, unfortunately, in our specific population, because the samples we had from 2017 also had spotty high depth full coverage at this region. So I think, because as I've actually spoken with Richard about this, sequencing that region is quite tricky. There's a lot of repeat regions, and sequencing quality in general is just fairly low. So this is why I think some long read sequencing would be fantastic to get for this region to actually confirm that this variant is there. And then when it comes to functional evaluation, I myself don't have any experience with doing this kind of research, but I would love to find out or work with someone who does have the ability to carry this out. It's be quite interesting, especially following Katie Coe's research is quite promising that there is more going on with these untranslated regions than we think there are.
So do you know of any other studies that have done this functional evaluation, or is it still to do then?
So yeah, it's still to do. Well, the issue is that the most relevant one that I mentioned is in Vivax, and doing that transcriptional work in Vivax is quite tricky considering our inability to culture long term. So that is something that would be quite, so something better, I guess, with working with falciparum in this instance is that we can actually do these transcriptional studies and get this functional analysis. In order to prove this otherwise, with the Vivax paper, they've had to go through quite a lot of work to kind of just, it's still not 100% that they can show this functional evaluation, but it's very strong. And so it should be feasible with falciparum.
And so you mentioned that the use of the antibiotic might also be causing some pressure on the anti SP mutations, but perhaps that this wasn't maybe it wasn't likely to be providing that strong of a pressure. Do you have any other hypotheses for what else, if not SP, and if not this antibiotic alone? Are there any other factors that might be at play here?
I wouldn't be surprised if these variants are playing a role in drug resistance on another level. So obviously we have the antibiotic pressure. I spoke with, I can't remember who it was. I was speaking to someone who is more working on the clinical side, and they weren't quite convinced that bacterial prophylaxis using this antibiotic would be high enough to keep that much pressure. And these variants would have already been there from 2004. The population's quite bottlenecked. Maybe there's no fitness cost to the parasite that suggests that they or warrants them to get rid of or drop these variants. So if they're not causing any functional or fitness cost to the parasite, why get rid of them? I think there's also potential that these drug resistance, they could be implicated in drug resistance elsewhere, but I do not have enough information to suggest that that might be the case.
So one more question from Richard. Do you have any hypotheses of why drug resistance genes are showing up more in IBD-based scans than in IHS-based scans?
I am not entirely sure. I was thinking that maybe it had to do with the window size we were looking at, or the way that maybe IHS is picking up more of these really strong conserved genes. I would actually really like to talk to Richard about why he might think that there's a difference between the two, because I understand why identity by descent would be a good way to look for regions that aren't recombining, and those regions that aren't recombining are therefore being deemed important or crucial to the parasites enough to retain those segments despite outcrossing and recombination with other parasites. But yeah, I've always kind of struggled to pick up drug resistance signals using the IHS metric, and I wasn't sure if maybe it was, yeah, I've consistently always struggled with getting those signals.
Have you, Richard, just written a
Yes.
follow-up? Maybe IBD-based might effectively combine signals from different mutations.
This is true. So that's one thing you might miss with the IHS-based metric, is perhaps the signal from one variant in a region isn't enough to pick up or be statistically significant compared to the rest of the region if that entire region is being selected for. So it will already be higher than the normal, but not high enough to be statistically significant. So that could also be the case.
So I'm just going to look through the Q&A. So I don't think we have anything there. Are there any more questions for Ashley from anyone else? Last one from Richard. Which do you think will be harder to eliminate from Ethiopia, Plasmodium falciparum or vivax?
I am going to have to go with vivax, I think. You're seeing, obviously, with vivax, you can have this dormant liver stage that's really difficult to treat and detect, and it's really difficult to implement and roll out the treatment with primaquine to effectively clear these liver stages because of the absence of like affordable and consistent G6PD testing. So without G6PD testing, it's not advisable to necessarily give out radical cure. Obviously, there are safe ways to do it, but there's quite a lot of hesitancy from clinicians and the community. And then another thing we're seeing is as falciparum is eliminated, vivax seems to be kind of taking its place. And so as that pressure from falciparum is diminishing, vivax is taking over, and then vivax has that dormant liver stage, which is really hard to keep track of, and you'll have asymptomatic carriers. I think it'll just prove really tricky.
And Sally Karimi has asked about, what about in Kenya? Do you think the same is true?
For falciparum or vivax?
I think this is the same question, but in relation to Kenya rather than Ethiopia.
Yeah, so in Kenya, vivax is not nearly as prevalent because of that absence of the Duffy negative phenotype, or because of the prevalence of the Duffy negative phenotype, sorry. So I think vivax is not really as big of a issue in Kenya, although I do think in Kenya there will become issues with the prevalence of malaria and ovale. It does seem in some regions where falciparum is diminishing once again that these other neglected malaria parasites are kind of resurging and taking over that. So although they might not be causing much clinical malaria like falciparum, it seems like just getting rid of falciparum might not be the answer, and there's quite a lot of hurdles to face.
Thank you. Vicky says, excellent presentation, Ashley. Thank you for your insights. So we've had almost 20 minutes of Q&A. I'm just going to give one last opportunity for any final questions, and then I think we might wrap this up. So I'm not seeing any more questions come in, so just leaves me to say thank you so much for giving us your time and presenting your work, Ashley. I really enjoyed that, and I thought that was a very interesting discussion too. Thank you everyone for attending this journal club. I believe the recording will be available soon. And yes, thank you and good afternoon.
Thank you, everyone, and thanks for giving me this opportunity. It's been really interesting, and the Q&A was also really kind of fun and interesting. So thank you.
Great. Bye, everyone.
Thank you.